right. Hello, everyone. Welcome to another Sagely Health webinar. Today is Friday, March 29th, 2024. I'm Jeremy, the CTO of Sagely Health, and I'm joined, as always, by Dr. Sager. Jason, how are you?
Fine, thanks, Jeremy, and hello, everyone. Glad to see you.
So we have had probably more interest in this webinar than any of our other recent webinars, and I'll be honest, we haven't been doing a lot of webinars recently. But the topic of cancer vaccines, which is today's topic, has been something we get asked about a lot. And it's something where I think we've had a measured response before. That might change a little bit. So Jason had said to me, we're very excited, I have a reason that I'm excited about cancer vaccines for the first time ever. And we thought, well, that's a great webinar. So Jason, before we get into your excitement and why you're excited, could we just talk a little bit about the confusion around nomenclature, meaning the naming of cancer vaccines? So everyone kind of understands vaccines as a preventative measure, not necessarily as a therapeutic or treatment measure. So could you just kind of explain what's going on here when we're talking about cancer vaccines in this context?
Yeah, absolutely. And again, it's understandable because the word vaccine is common to both the preventative vaccines that we get for COVID or basically or all of the childhood diseases and cancer vaccines, which is specifically being developed to try to get the immune system to attack the cancer. And so, the two are very different. And the basis of that is that the vaccines that are preventative are trying to attack viruses and viral proteins that then, infect the body. And so the body is preventing that infection by having the vaccine on board. In cancer vaccines, it's entirely different because the vaccine, quote unquote vaccine, and we'll talk a little bit more about what those vaccines are. They're really trying to get the immune system to attack a cancer. Well, cancer is a human, a cell in the body. And so you're relying on the differences between normal proteins and, let's say, altered proteins that the cancer has that the normal body does not. That's much harder to do than looking at virus proteins and trying to reject those from entry into the body. And so at the end of the day, even though the vaccine is the same, they're really quite different entities. And unfortunately, you can't mix them. This comes up because one of those cancer viruses, human papillomavirus, HPV, can cause cervical cancer. And so the question is, well, would a HPV vaccine be helpful? And the answer is, well, if you're doing it before you get infected, then yes, because if there's no virus, there's no potential to become cancer. However, once it's become cancer, it's not useful to get that antivirus vaccine. That's sort of the virus has already created the cancer, and now you need a cancer vaccine. And so really trying to keep the two separate is very important, but there's lots of reasons why it's confusing. Today, we're going to be focused in on those cancer vaccines, and we'll talk a little bit more about that.
Okay, so just to be clear, there is no vaccine that we have today that prevents cancer. There are vaccines that prevent viruses that could cause cancer, such as preventing HPV so that you don't get HPV. So there's no chance of that turning into cancer. And so we're not talking about prevention, except for when we're talking about something that's for viruses like HPV. So the reason they've said vaccine, and I think I heard you say, is that it's about attacking something, getting your immune system to attack something, and instead of it being the protein of the virus that would keep the virus from kind of taking host, we're talking about a different type of protein that's in the tumor.
Right. And we're looking at those proteins that are altered. We know that cancer is a disease of genetic alteration, so the DNA, and we've reviewed this otherwise, is altered, has mutations, amplifications, other things that are different, that make it different than the normal cell. And as such, it then can stop acting like a normal cell and start proliferating and forgetting about where it's supposed to belong, and thus it becomes cancer. So these vaccines, the idea behind them is that those proteins that are different, and pieces of protein have something that we can utilize to try to excite the immune system to then attack. That piece of the protein we call a peptide. A small piece of protein is called a peptide, and the ones that we think may be helpful in attacking it are what we call neoantigens, which is new antigens, new things that can stimulate the immune system to attack. And so you'll hear those terms a little and see those terms a little bit later when we discuss the paper that we're going to present. But those are the fundamentals behind understanding what a cancer vaccine is designed to do.
So, so this falls into the broad category of immunotherapy, meaning we're asking the patient's immune system to kind of step up or be stimulated by an intervention. So, could you talk a little bit about kind of the history of cancer vaccines, kind of what has led us to this point?
Yeah, there's been great excitement and enthusiasm for immunotherapy, and much more recently, within the last 5 or 10 years, by the virtue of the discovery of what they call checkpoint inhibitors, such as PD-1, PD-L1 as a target. That is different. That's sort of like a cloaking device, and again, we review that elsewhere. Cancer vaccines are actually even much older, where folks were trying to grind up tumors and find the proteins and just inject those proteins to see, could we stimulate the immune system in a way that would then reject a patient's cancer. Logically makes sense that that might be able to happen. Hasn't worked. There were others who said, well, let's try to analyze the tumor and do sequencing of that tumor, which is great to find avenues by which to attack it, but then to use the analysis of that to then educate, create peptides, and then utilize that to excite the immune system. Up until recently, up until now, that also hasn't been met with overt success. One of the nice things about it is that these small pieces of peptides that are used, these small pieces of protein that are then being injected into someone's arm in order to stimulate the immune system, there's very few side effects, like none, right? And so from a safety perspective, and as a cancer patient, you're living in a world where most of the medicines create side effects, and some of them quite significant. So to hear about a potential of something like a peptide, a vaccine that could be injected in my arm, and I'm not going to have any side effects, maybe a little arm, soreness, great. On the other hand, it also hasn't worked that well. And by virtue of that checkpoint inhibitors, which has worked amazingly well for melanoma, for lung cancer, some places in breast cancer, but has nothing to do with those vaccines, the world is starting to ask, can these two things be put together in a way that's more successful? And multiple companies have worked on this, multiple scientists, academic centers, and now for the first time, I'm excited because up until last week, I hadn't seen real evidence to say that they were working. I now have. And what's interesting is it's coming from a place of great unmet need, high need, which is specifically pancreatic cancer patients. And so, I know we have many pancreatic cancer patients of ours out there and folks who are interested. And certainly that it's an area where we all want to be doing better. And so I was really encouraged to see this, even if it's not for metastatic cancer patients, it's for local, or, and we'll discuss a little bit more about what the criteria were to enter the clinical trial for this. And the company is BioNTech. They're known because they do produce the COVID vaccine with Pfizer. That being said, again, not related to that. This is the cancer vaccine, and but it is a real company, and I think they're going to be able to do more with their initial findings, and with the hope that that it will expand. And I'll show you a little bit of that today.
Yeah, I mean, I want you to go into. I mean, my biggest question is what changed, right? Like, if this has been around for a long time, what
Yeah.
has happened that allows us to kind of see better results?
Yeah, it's a great question. And again, from my perspective, there's two sides to it, right? The first one is, remember that avenue of those checkpoint inhibitors that before 5 or 10 years ago weren't even discovered. And so you have this avenue. And for the first 5 or 10 years, they were just using it by itself because in melanoma, it worked really well. We're now at the point where we're using it enough that they're now starting to combine it with other ideas. And this is a pretty obvious one to combine it with. So you'll see in this paper, in this trial, that actually they did put these two together in a very specific way. And others can learn from that and say, okay, well, let's try the same thing with our way. The second thing that's changed is the way to analyze those proteins and which peptides you make from them and how those are made. And so that is the second part that I think is novel or potentially novel. And so we'll see, and there's some secret sauce there. So I don't know that we know exactly how BioNTech did it. I'll show you what they showed us. But in the sense that others will have ways of looking at this and either doing the same or slightly differently, but to do it in more robust ways. And so again, to me, we're not at the point where I'm ready. We're ready to say, okay, we're done with this. But it is the first spark, I'd say, of interest and real evidence to say there's something here that wasn't here before, and that is worth talking about and having much more effort now going into in order to see if we can get it to work for more patients across multiple cancer types and in all sorts of stages.
Yeah. So what happened here with
Yeah.
pancreatic cancer?
Yeah. So just to show, I'm going to project my screen here, and just to show, this is the article. It comes from Nature, which is not surprising because they often publish very landmark and very interesting studies. There's a lot of various authors on this, some of whom I very much recognize. Eileen O'Reilly, for example, and others who have, again, been working their whole lives in trying to do this. And so it's great to see Jed Wolchek and others on this paper. As we go through it, and I'm gonna obviously skip to some specific things that I wanted to show all of you. Let me just say that it's complicated science, and the amount of science that they show here is, I would say, extraordinary. That being said, in terms of discussing it, I wanted to home in on a few of the key points. And here I'm showing a little bit about how they did what they did. And so they took patients who had pancreatic ductal adenocarcinoma, which is the common type of pancreatic cancer. These were only patients who had surgically resectable, local, meaning small enough tumors, where surgery would be expected to take out all of the cancer. Now, even in those patients, unfortunately, we know that they're at high risk, that the majority of them are going to get cancer back again. And I'll show you the data behind that. At the same time, they took patients who, and these were patients who were also newly diagnosed. So they had not yet been treated. They then were screened for eligibility. They then had surgery where they had probably a Whipple's, where the pancreas was removed and the tumor was removed. They also took some blood, and they then compared those and analyzed that tumor in blood and fed that into their system to predict and select those neoantigens, those small parts of the protein, which they felt like could excite the immune system.
Is this the
From
part
there,
that you're saying kind of is their secret sauce?
yeah, this would be the secret sauce right here, although there's also maybe some sauce here as well, but certainly this is the prediction part of it. And other companies like CGAT are doing great work with this. There's other companies that do this. Again, these are the first folks that are publishing in a way that, to me, say something is successful here, and I'll show you that in a minute. From that point, they then produce those small custom peptides, those small pieces of protein, and put them into a bottle. And then they then deliver it to the patient. And you can see here that they had the surgery. 6 weeks later, they get a dose of this atezolizumab, and that's one of those checkpoint inhibitor blockades. Pembrolizumab, nivolumab, Keytruda, Opdivo. These are all names, and there's I think 8 or 10 of these drugs out there now, and they all kind of work similarly. So they're getting a dose of this checkpoint inhibitor, again, to kind of prepare the immune system It's not going to be able to cloak the tumor like PD-1 usually does. And so they're combining this approach. And then they start giving the peptides when what they call the name of their product is autogene cevumerin. And they start giving at week 9, and they give several what they call priming doses until week 17. Now at week 21, they start some chemotherapy, and they're using the standard FOLFIRINOX, modified FOLFIRINOX, and they're giving that for 12 cycles bi-weekly. So another 6 weeks go by, or actually more, no, every other week. And so now we go from week 21 to 43. And then they're giving a booster of the vaccine at week 46. And then they're watching for follow-up. And so again, these are patients who have the tumor completely removed. These are patients who are having adjuvant chemotherapy, as patients in this situation would have, but they're also getting that checkpoint inhibitor, and they're getting this peptide vaccine. And so that's sort of the first sort of, how did they do it? And this sort of answers all of that question. And let me just pause there and ask if there's any questions on that. Otherwise, we'll start talking about their results.
Well, my question is I think you touched on this, but just to be clear, that if in the participation in this trial, they got this treatment, what they normally would have gotten outside of the trial, the standard treatment would be this Whipple surgery if they are eligible, if it is resectable, and then having the adjuvant, the therapy after that. So what we've just done is insert also the kind of the dosage of their vaccine, and then the follow up following the chemotherapy.
Yeah.
So
right.
So the study compared people kind of didn't have that and those who did, right?
No, actually.
No. Okay.
Yeah. So they actually did this to all of the patients. And here on the right, you can see that they ended up screening 34. They ended up treating, they have 15 that basically they were able to evaluate from that.
So whatever numbers come out of this, you have to kind of apply to what is the standard results, right, without
we have a little bit of benefit here. And the first thing that they saw, and now, again, they included a lot of information, which is great. But really, to me, what is most important is actually just this red-blue circle, 50-50. Half of the patients they saw responded, so they were able to see the immune system get activated in half of the patients in a way that was specific to the peptides that went into the patient. So they could actually measure this in the blood of the patients. The other half of patients did not respond. And so really they had their own control. I don't think they designed it this way, but they happened to find that this was what they found. And so they have half of the patients, 8 patients that responded, and 8 who did not. And so now the question becomes, well, how did these 8 do? And how did these 8 do? And we can compare it to each other by the virtue of, even though they've all gotten the same exact thing, half of them had an immune response and half of them did not.
Well, my first question is kind of why? Why would someone have an immune response and why would someone not?
So let's come back to that. Let's come back to that. I think
prior
to today, no one's bothered to ask this because everyone's not done. I mean, there was no difference between they both did equally not well. And so, okay. One question we were asked, though, just as I go to the results, I was asked, like, well, does this apply to pancreatic cancer patients? The answer is yes. Many pancreatic cancer patients have a mutation in KRAS, oftentimes G12D. There's many others, but KRAS is very frequently mutated. That would go into their neoantigens. So the fact is that, yes, like this is for KRAS. This is for basically any of the P53, any of the proteins that get mutated can go into this complex algorithm of producing these neoantigens. So the answer is yes, it's for those patients as well. I know we were asked that.
That's a really important part. Like the top of this article was, personalized cancer vaccines. And that's a really important thing to say. There's a procedure here. Things are removed from you. They're training. They produce things. They give it. So that one cancer vaccine from them doesn't fit all. The process does.
Right. Exactly. If you had a PI3 kinase mutation, then you'd get a peptide against that. And some of them don't even make so much sense, but they see that there's differences and they can utilize that to teach the immune system. So again, this is just the spark. There's so much more learning we're going to need to do. And again, as they presented some of their information, and now I'm scrolling down, like, they show a lot of this characterization of the immune cells, and they're trying to understand it and what's going on with the responders versus the non-responders and all kinds of stuff. I want to skip all of that, and we'll let the scientists do what they do. But at the end of the day, I wanted to bring you to this their findings of outcome. How did these patients do? Remember, we said that they all got the same treatment, that checkpoint inhibitor, and then the vaccine, and then the chemotherapy. Half of them responded to the vaccine in their immune system. Half of them did not. When you look at how much time did it take? Did their tumor progress? Did it come back? All of these patients had their tumor completely removed. So did the tumor come back? And here you can see that in the red are the 8 responders, and in the blue are the non-responders. And what you could see is that over a period of anywhere from about 8 months or so till about 30 months, these patients were monitored. By the end of it, I'd say all except for one patient, perhaps, had their tumor come back if they were a non-responder. Of the responders, none of the tumors came back. And so this is where we say, oh,
Yeah.
something is going on here. This is very clearly different. And it's not like, they're close together and maybe one's a little better than the other. No, 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 no. This is dramatic, right? And even though there's small numbers, only 8 patients, this is a spark. This is a true spark where none of those patients had their tumor come back. That can't be by chance, where everyone except for maybe one of the patients had their tumor come back in the other group, 7 out of 8. That just cannot be chance that just randomly happened. And so, and actually they have their p-value is the way of scientists measuring that, and it's highly significant, 0.003, 0.5. So 50 would be the comparator to that, to say it's not significant. So it's very, very significant. And so so this is where Nature and the rest of the world say, wow, this deserves publication. This deserves to be on the front page of the New York Times. And even if it's not, we're going to try to put it there.
Yeah, so because that blue line, though they were given the vaccine, they didn't respond. That basically represents a normal patient's kind of path before this.
That's correct. That's
Surgery,
correct.
and the chemotherapy
Yeah.
following surgery, and you see you get 4 months in, somebody had a recurrence. A little
Right.
later, someone else had a recurrence.
And about half of the patients recur after a year, which is pretty much what's been known as the standard for pancreatic cancer patients, unfortunately.
So pancreatic cancer, it is a huge, like you said in the beginning, an unmet need, just because the outcomes for pancreatic cancer are not good. I mean, in the sense that the cancer does return if surgery is possible in so many of the cases. So we did have, I think you might have other things, but I did want to jump into the fact that we had questions from people. And a lot of the questions were around, does this apply to my cancer? And will it apply to my cancer, for everything from triple negative breast cancer to ovarian cancer, to other cancers? So how are you thinking about that? I mean, because we're looking at one trial and this trial concluded, and I know that they've started other trials to continue studying this. So should patients kind of expect this to be available across many cancer types in the future?
Yeah, I mean, again, our objective is to try is if it is available, to make sure we can find, help patients to find out where it's going on nearest to them, and get access to that by virtue of getting a second opinion, meeting the investigator who's running the trial at the cancer center. So let's say Dr. O'Reilly at MSK, we send the patient in for a second opinion there, and then with the idea that, hey, I'm interested in this vaccine trial, and that's the way to do it. I think that the cancer world takes a while to do this, and we'd like to accelerate that. So as much as there is interest and enthusiasm, you know, approaching BioNTech, letting them know how interested you are in this, and letting your friends know as well, other cancer patients, so that we can get those trials as quickly as possible. So this becomes an approved therapy. And once it's approved, then it becomes much more available to the masses, to every cancer patient for whom it
Right.
fits. And so there is sort of that, not bottleneck, but sort of this process of going through, because they're still evaluating how safe it is and how effective it is. And remember that, most drugs are not as safe as this. So that's where the system comes from. It can go a little faster here, but they'll still have to do their phase 2 and 3 confirmation trials and apply for approval for it, before it's available sort of to every oncologist to give to their pancreatic cancer patient or their colorectal cancer patient or whatever, specific cancer type that they've studied. And so, it's a process that's going to take some time, but that's where we're able to help patients access the future of medicine, these things today in trials. And so that's what we do.
Yeah, I'm struck by the fact there are eight patients who went on this trial who are going to have a dramatically different outcome, at least as far as 30 months in there. We're looking at nearly, I mean, and I'll be interested to see how long that red line stays right there at the top in what you've shown. And I think that's what we kind of see for patients is if you want to know about whatever the innovative options that are available are, that's why we're here. I mean, because you
Yeah.
will get the standard treatment, through your oncologist. That's what's available. But we're looking at the data as it comes out, and this is something where, like you said, like weeks ago, before you were looking at this data, the cancer vaccine trial might not have gone to the top of the list. But now for certain patients, this definitely does.
Yes, yes. And I'd say similar trials that are doing this approach in combining those checkpoint inhibitors in the right way will also gravitate up. And so, again, there'll just be more renewed interest in all of this. But yes, I think the greatest expectation is that this company with this approach is going to mean more success. So let's fill up that trial and let's get those patients some benefit as we can.
For sure.
I,
So,
I do
okay.
want. I do want to. I want to. Answer one more question that we were asked about going back to again. Right now, today, this was for patients with local, and unfortunately, a lot of patients come in with already metastatic disease. And so, does this approach really have usefulness for that? And part of the idea here, and the reason they started is theoretically, you need to give the immune system some time to learn. And if the cancer is actively growing, it's sort of like a race. And in trying to stack the deck in our favor, that's why they started with patients whose tumor can be completely removed. But something really interesting happened, and I wanted just to again show one other. It's a little wordy here. It's a paragraph, but there is a patient that they spoke of, patient 29, and I'm sharing my screen again, who responded in their blood to the peptide vaccine, um, and they had the second highest maximum response. Okay. What was interesting is that all of a sudden that patient's tumor marker called CA19-9, it's a common tumor marker that many patients get with pancreatic cancer, started to go up. And you say, uh-oh, this is a cancer that's coming back. Indeed, they did an imaging study and they saw a new 7 millimeter nodule suggestive of a metastasis. And this could be that could wreck the that line for sure if this were to be proven. Now, they went in to try to do a biopsy, and sometimes those biopsies can be really hard to get a very small tumor like a 7 millimeter tumor. And they didn't see any tumor in that biopsy. But what was interesting is, but they did do some other tests, and it looked like that there were other signs that this tumor was coming back. They continued to follow the patient, and here's where the sort of the miracle happens: is that the liver lesion, that nodule, disappeared on subsequent imaging. And again, there's no absolute proof, but there's an absolute suggestion, and I'd say highly likely, that the immune system attacked that tumor. Oh, that's one of the things that they saw when they actually looked at the biopsy. They didn't see tumor. They saw a bunch of immune cells
Oh, wow.
as if they were attacking the tumor. And those immune cells were specific to the mutation that the tumor had. The P53, we talked about that, this R175H. And so, again, early signs,
Yeah.
but gosh, this is a patient who almost, again, was going the wrong direction, and all of a sudden, and again, this is not the way things usually ever happen. And so not in pancreatic cancer. And so again, some real reasons to be excited about this and the future, that this could be eventually effective for metastatic patients, because they certainly didn't aim the immune system at the liver. That's the beauty of the immune system, is it can find cancer cells wherever they are and attack them. So I think there's great future potential for this therapy.
That's incredible.
Yeah.
Wow. All right. Well, might be a good place to wrap up. Anything else that you want to add?
No, I think that's it. Again, we're always here for helping to guide patients, finding the treatments that of the future today, that will both increase their longevity as well as their quality of life. It's our mission, and it's what we do.
Yeah, I just say, Sagely Health is a cancer guidance service. We have some deep technology that helps us find solutions for people, help them get them access. So if anyone watching is facing a cancer diagnosis of any type, not just pancreatic, but any type, or a family member, please reach out. We'll want to make sure that you get plugged into the best possible treatments for you. And we're constantly tracking everything that is going on. So. Well, thank you, Jason. This has been a great conversation, and now I think I share your excitement because I understand it a little bit better.
Yeah, excellent. Well, looking forward to more. Thanks so much.
All right, thank you. All right, thank you all.